Hello, everyone. My name is Austyn Trull, and it is with great pleasure that Dr. Ladayanov and I will be presenting the work that we've done on the NF-core scnanoseq pipeline. So scnanoseq is a pipeline that performs secondary analysis on long-read single-cell RNA data. We've developed it so that it uses the best practices introduced in the single-cell and single-nuclei space, as well as ensuring that it is reproducible and portable. So getting into the pipeline overview, as I mentioned, we do utilize the NF-core framework to develop the pipeline, which does mean the pipeline is written in Nextflow. Because we do use the NF-core framework, we are able to leverage both Docker and Singularity to ensure reproducibility and portability. As far as inputs, we do expect fast queues. These fast queues need to be 10x barcoded, as well as processed on a nanopore machine, so a GridION or a Prometheon. Kind of quickly going over the steps. I won't list the tool, but they are listed in green. So we do read filtering, barcode detection and extraction, alignment, barcode correction, barcode tagging, UMI deduplication, gene and transcript quantification, and then we also do QC at various points in the pipeline. So kind of getting into that, what the pipeline actually produces is we produce fast QC reports on differently processed fast queues. As the pipeline goes on, we produce nanocomp reports on the raw fast queues and the initially mapped VAMs. We also produce the detected barcode files produced by Blaze. We produce alignment files at various steps. And along with these, we have various SAMTOOL stats run on each VAM. The final output of the pipeline is, of course, the gene and transcript level matrix. And in addition, we do run preliminary QC using Seurat on the matrix. And then finally, we produce a multi-QC report that kind of summarizes and includes some of the various QCs the pipeline runs. So we do have examples from an example run, both to the right and below of this text. So you can see right here some of the fast QC or fast Q quality control images, which includes nanocomp and fast QC. We include tables and images for BAMs, which some of these are the SAMTOOL tables, as well as, again, nanocomp to view all of your samples. Then finally, we do include some single cell QCs. So you can see we do have a table listing some of the cell numbers, the mean reads per cell, and the median genes per cell, as well as a knee plot and the preliminary images produced by Seurat. So with that, I'll hand it over to Dr. Ladayanov, who will go over the validation for the pipeline.
Hello, everyone. I'm Dr. Ladayanov, and I'm picking up where Austyn left off to cover the validation section of our poster presentation. So the goal here was really to reprocess a published data set with scnanoseq to demonstrate to you that the results generated by scnanoseq are indeed biologically meaningful and are valid. So the entire section is really to compare the original published data set versus what we see here. So to jump right into it, panel A displays the scnanoseq data at the top at the gene level and the transcript level, and at the bottom I have the ground truths or the published data shown at the gene level and the transcript level. And as you can see, we are able to indeed reproduce those same 6 clusters of cells that they have identified in an almost exact fashion. Panel B is a similar plot, but with the UMI counts. And once again, you can see that we get a similar profile from either the scnanoseq at the gene level and the transcript level versus the ground truth data. And we see also in this particular section that we are able to recover slightly higher UMI counts, which suggests that the results from scnanoseq could be more sensitive. Panel C and D displays the gene expression of specific key genes that the authors chose to highlight in their paper. So we've chosen those same genes here for you to be able to see. And overall, as you can see, the profiles are very similar, if not the same. And in some cases, such as PAC6, it does suggest that the scnanoseq results could actually be slightly more sensitive. Panel D is, again, the same information, but at a violin plot. Panel E and F is that data, but at the transcript level, and that same story continues. We're really showing that we are reproducing the original results with some slight difference in sensitivity. Panel G shows a correlation between the 2 data sets with scnanoseq shown at the x-axis and the ground truth shown on the y-axis, again at the gene level, the transcript level. These two figures displays the correlation of the number of genes between the two data sets, and these show the number of molecules. And as you can see, the correlation is very high, specifically with a Pearson correlation of 0.95. And last but not least, panel 8 displays the barcode validation of the ground truth versus different outputs from scnanoseq, and we indeed get high intersection of these barcodes. So hope this has been enough to show to you that we are able to process this with high accuracy. And if you'd like more information, please contact us specifically at the NF Core channel. That will be our preference, the scnanoseq. So we hope to see you there.